Start the journey. So as Joe said, she wanted me to introduce how I got here, and as you can see from the first slide, that was not a straight path by any means. Can everyone hear me? Okay. Okay. Great.
Good.
Okay. So I grew up in Lee County, which is the very western tip of the state of Virginia, in a little town called Jonesville on a small farm. Those of you that are history buffs may know that that's about 35 miles from Cumberland Gap, which is where Virginia, Kentucky, and Tennessee all come together. And that was America's first gateway to the West, where Thomas Walker led the first settlers. Daniel Boone's son was killed about 20 miles from where I grew up. So when I was 5 and my sister was 7 and my younger brother was 2, we lost our father. But we were very fortunate to have a mother who really completely dedicated her life to raising 3 small kids. So when I was entering my senior year in high school, although it was a hardship for her, she insisted that I go to college. And since our family was of modest means, our guidance counselor suggested that I should apply to Berea College. For those of you that may not know, Berea was founded in 1855 by John G. Fee, who was an abolitionist, who thought that blacks, whites, men, and women should all be able to get an education. Berea charges no tuition. Everyone has to work at least 10 hours a week. And you can make enough money with your room and board, with your labor earnings to pay your room and board. So you can essentially go to college for free. And actually, to this day, Berea still charges no tuition. So after graduating from Berea, I was fortunate to get into graduate school at the University of Tennessee. And, that turned out to be an amazing, exciting time. I was getting my degree in microbiology, but it was really microbiology that gave birth to what is now modern molecular biology. Because the first restriction enzymes, which are enzymes that are made by various microbes, were the tools that allowed us to be able to cut DNA very specifically to initially clone fragments of DNA. So that was really the beginning of molecular biology. So after I completed my Ph.D. at the University of Tennessee, I was able, first, the best decision I ever made was to marry my wife, Betty. And if you look at that date and do some quick math, you'll realize that Betty and I actually just celebrated our 50th wedding anniversary this past Saturday. So Betty was a graduate of the nursing program at Berea, and she has been a very supportive spouse and partner through this last 50 years of journey. And so I would suggest to all of you, the most important thing you may ever make a decision on is the partner you pick to spend the rest of your life with. So we then moved to Houston, to Baylor College of Medicine for a postdoc. And again, that was a very exciting time because we were working. I was fortunate to work with Bert O'Malley, who was my mentor. Bert and his group were racing to see who could clone the first genes, and we were racing against Pierre Chambon's group in France. So when I was, that was a very different looking me when I was at Baylor during the late 70s. We made a lot of friends at Baylor, and we learned a lot of Texas stuff, as we say, like dancing the Cotton Eye Joe. It was a great time, very exciting, a lot of postdocs all the same age, and so we had lots of energy, and Bert really ran his lab as a work hard, play hard operation, and so it was really truly a great time to be there and to be in science. We not only played, but we actually did get a lot accomplished. Was able to publish a few good papers. And I thought I might just show you a little bit of science that some of you might find like it's really from the dark ages now. But when we cloned the first genes, we were trying to figure out, so Chambon's group published the first structure, but we were the ones that actually published first how the genes were actually expressed. And we were racing with Phil Sharp's group. Phil Sharp ended up beating us by publishing how one of the adenovirus genes was actually expressed. He ended up getting the Nobel Prize for that. But what we discovered was that the genes actually had large, what we now call intervening sequences, or introns. And when we ran, if you look on the left, that's what we call a northern blot, where we extracted total nuclear RNA. We then used a probe that was just for the coding sequences. And you could see all of these bands. The RNA is separated on the basis of size. So the largest band at the top is the largest molecular weight. And the mature messenger RNA is shown down at the very bottom. So you could actually see that these were precursors of very large transcripts. And to understand the structure, we actually hybridized the messenger RNA back to the full-length gene. And the loops that loops out, as you see in that electron micrograph, were actually the introns. So the only sequences that the coding sequence for the messenger RNA would bind to were the little exons. And so that was the first paper we published to show the exact structure. And that's a schematic of the structure. So the little dark boxes are actually the exons, and the large white boxes are actually the introns. So it was really amazing to realize that these genes were actually much larger than we ever anticipated. And they were actually transcribed as a very large precursor, and then the introns were spliced out. So that's a little bit of the science that we did when I was a postdoc. After leaving Baylor, I was recruited to the National Cancer Institute by Stu Yespa. When I went to the NIH, I knew nothing about the skin. I was a molecular biologist. We called ourselves gene jocks. We knew about DNA and we knew how it was expressed, but as far as skin biology, I knew nothing. And that's Stu's lab worked on skin biology and skin cancer. And at that time, we knew very little about the proteins that were actually expressed in the skin because they were all insoluble. So traditional biochemistry that had been used to solve the structure of soluble proteins just were not effective with the proteins expressed in the skin. As you can see, I was fortunate again to work with a mentor who also loved having a great time. We still are really great friends. Stu still is working. It's amazing. And when I was president of the Society for Investigative Dermatology in 2004, I actually was able to give the Rothman Award to Stu, which is the highest award that the society gives to a scientist that's made the most outstanding impact on skin biology. So basically, I won't get into the science, but we ended up essentially cloning every gene that was expressed in the skin when I was with Stu, and we sequenced the DNA structure, and that revealed then the protein structure. So when I was recruited then back to Baylor College of Medicine as a faculty member in 1988, we had the structure of the gene, we had the structure of the protein, but we really didn't understand what the proteins did. We knew that they were structural proteins and they were expressed at high levels in the skin, but we didn't know exactly what they were doing. So, this is just again a fun time. This was the first lab party we had when I was recruited back to Baylor. I took several people that were working with me at the NIH with me to join me at Baylor. And so again, great times, lots of fun times at Baylor. We did enjoy Halloween, and Bert O'Malley, who was the chair of the department, loved Halloween contests. And so we would have theme contests for each lab group. Here you can see we're the Texas Killer Bees. Here's another one where we are the serial killers. Again, a lot of very imaginative individuals that I had the pleasure of working with. So just to talk a little bit about the science, and this explains how I got to where we are today. So since we didn't know exactly what the proteins did in the skin, we decided to take a reverse genetics approach, where we actually mutated the genes, introduced them into mouse embryonic stem cells, and then produced mice that had the mutated genes. And the first thing that we saw was these mice developed blisters on their paws just from walking around and the bedding in the cage. I was fortunate to have a dermatologist working with me, Daniel Ho, who's shown here in this publication. Daniel Ho was trained in Switzerland as a dermatologist. And as soon as we looked at the histology of the blisters, he said, I have seen that disease in humans. And so he contacted his mentor in Switzerland. We were able to obtain DNA from one of his patients. We sequenced the DNA and found a mutation in one of the keratin genes. And I'll explain how that works in just a few minutes. We wanted to confirm the father actually also was affected back in Switzerland, but he lived in a very remote mountain region and did not want to come back into the city to the clinic. So we were actually able, at that time, and I think we may have been the first, we actually got a histology slide of the father's skin tissue. We actually isolated DNA from the slide and confirmed that the father had the same mutation as the son. And we ended up publishing that in Science, and that was the first report of these particular mutations causing this skin fragility syndrome called epidermolytic hyperkeratosis. So these are just examples of kids that are born with mutations and keratin genes. On the left is a disease called epidermolysis bullosa simplex. And on the right is epidermolytic hyperkeratosis, the disease I just mentioned a few minutes ago. I know these slides are difficult to look at, but I've said this many times with my trainees now, and we really insist that all of our trainees have clinical co-mentors. Because I think once you, as a basic scientist, if you're fortunate enough to discover the genetic basis for a human disease, and then you see the phenotype that these mutations cause, you'll never look at the way you do science again. So I think it's really important for basic scientists to actually meet the patients, meet the parents, and it will impact you forever. So as I told you, we were able then to deduce the structure of the proteins after we cloned the genes from the DNA sequence. And so this is kind of a stick diagram that shows the structure of a keratin protein. And you'll see that there are different structural domains, but on each end, we call the region on the left, the head domain, and the region on the far right, the tail domain. And we know that there are actually hotspot mutations that occur in these regions. So we know that in order to form a keratin intermediate filament in a cell, which is shown in the bottom there, that nice green um, wavy scaffold, if you will. So this is a structural filament that's formed on the inside of the cell. In order to form that, we need the product of two different genes. So there are two different keratins that form a dimer. And then for these proteins to assemble into a large filament, the head and the end domains have to interact. And those are shown with the red and green balls. And so the yellow ball then is where we have a single point mutation. And when that occurs, the filaments can no longer form long filaments. They form short, stubby filaments or aggregates. And in that way, the nice scaffold that forms on the inside of the cell completely collapses. So basically what happens in this blistering disease is the individual cells that are fragile and they just blow apart in response to mild mechanical trauma. So these are, we heard from Ella a little bit about dominant mutations or gain of function mutations. So these are dominant disorders. So a mutation in one gene produces a mutant protein that actually interferes with the ability of the other protein to function. So if you look now, we know that there are over 40 different genes encoding structural proteins that can cause different types of skin blistering disorders or fragility disorders and other epithelia. The ones I just mentioned that are shown at the very top there, EB simplex, those are dominant disorders. They are intracellular proteins. But if you look at the bottom, we see laminin genes, we see collagen genes. These are proteins that are actually secreted outside the cell. And these disorders are caused by the loss of expression of both alleles. So they're recessive disorders. And so like with type 7 collagen, that's a protein that anchors the epidermis to the dermis. And so when you don't express type 7 collagen, the epidermis just peels off. So these are the different types of fragility disorders that can cause these inherited skin blistering diseases. So in 2007, I was recruited from Baylor after 19 years to the University of Colorado to establish the Gates Center for Regenerative Medicine. And when I left Baylor at the last going away party, Bert O'Malley, who was my mentor as a postdoc and then my chairman for 19 years, when I told him about the position, he told me, he said, if it's half as good as I've heard, you've got to take the job. Go up there and make us proud. So again, you know, as you go through your careers, I would encourage you never burn bridges. Always leave on good terms. I'm still very good friends with Bert and Stu Yaspa, mentors that I was able to train with. And I think it's really important that you try to do that with your own careers. So when I went up prior to arriving in January of 2007, I went up for a press conference in August of 2006. This is Diane Gates-Wallach. She and her brother John are the founders of the Gates Center through the Gates Frontiers Fund. They made the initial gift that allowed us to leave Houston and move to Colorado. So at that press conference, I actually stood up and said, we're not going to work with embryonic stem cells. We're only going to work with adult stem cells. And at the time, that was kind of like a fighter going into the ring with one arm tied behind your back, because it looked like all of the potential was with embryonic stem cells. But I think you'll know there were issues with embryonic stem cells, both ethical and political, as well as potentially biological reasons as well. If you remember, George Bush had actually vetoed twice federal funding for use of human embryonic stem cell lines. So if we'd even wanted to work with human embryonic stem cell lines, we would have had to have done that in a separate facility where we didn't have federal funding, and that was just not feasible. Also, coming to Colorado, I was going to be fundraising, and I didn't want to start on a controversial note. So again, another fortunate thing that happened was literally a month before I came up for the press conference, a Japanese scientist, Senya Yamanaka, made an amazing discovery that you could actually reprogram adult cells back into an embryonic-like state. And so that's shown in this schematic, at least in the case of human, where you take a small biopsy from the skin, put the fibroblasts, which are present in the dermis, in culture. You then add 4 reprogramming factors, and over a period of about 2 weeks, you can reprogram an adult committed differentiated somatic cell back to a pluripotent embryonic-like state. Now that was just an amazing discovery, and I'm sure I wasn't the only one. If you had asked 100 scientists, 99 would have said there's no way you can do that. But it actually turned out to be a very robust technology, and it was actually fast-tracked. And so Senya Yamanaka and Sir John Gurdon actually shared the Nobel Prize in Medicine in 2012. So basically, Yamanaka's work was really based on seminal studies carried out by John Gurdon almost 40 years before. And they're shown schematically here. So John Gurdon worked with frog oocytes or frog eggs because they were large and you could manipulate them. So you could actually, you could remove the nucleus and you could transfer a nucleus from an adult cell into an oocyte or egg. So basically, this is what he did. He took a skin somatic cell that was a fully differentiated adult cell. He removed the nucleus from that cell, and then he removed or destroyed the nucleus in the egg, placed the nucleus from the adult cell inside, incubated it, and it became a frog. So what that basically said was, if you think about normal development as the embryo starts to differentiate and specify into different adult lineages, for example, into skin or to bone or to muscle, you don't want all of the genetic information that's accessible and available in a pluripotent embryonic cell to be expressed. You want to really shut down and lock down and only express the DNA that you need to form another skin cell or another muscle cell. And the way that nature does that is you basically lock down the structure of the DNA or the chromatin in a locked configured state. So it's kind of like taking a lot of files on your computer and encrypting them so they can never be opened back up again. Basically, what happens when you transfer that adult nucleus into the oocyte, there's something in the cytoplasm of the oocyte that can actually unlock the encryption. It really takes all of the negative marks that shuts down the chromatin in the adult cell, opens everything back up. So now all of the files are accessible. All of the genes can now be expressed. So that's really basically what reprogramming is all about. So if you are interested, I would encourage you to go listen to the Nobel Prize lectures that John Gurdon and Yamanaka gave. They're very lay friendly. And when Gurdon was giving his lecture, he actually showed this image. And he said that was the first frog that he actually developed by nuclear transfer. And as he said at the time, it was a very good frog. It lived for 20 years and produced 30,000 offspring. So that was really the basis that suggested that you might be able to actually identify these factors that could actually reprogram an adult cell back into an embryonic state. So that's exactly what Yamanaka did. And it was really trial and error. He basically took all of the genes that were expressed in an embryonic cell, cloned them, and then in different combinations started transfecting them into, again, just a somatic skin cell, a fibroblast that was fully differentiated. And, you know, it took years to find the right combination. He had several of his trainees that left the lab. He almost gave up himself. But then he finally found the right combination. And the right combination was 4 factors: OCT3/4, SOX2, c-MYC, KLF4. And so he initially published that in 2006. And then within a year, 4 independent groups had actually reproduced it also in human. So it turned out to be a very robust technology. And this is really was the game changer for us that allowed us to be able to compete without using embryonic stem cells. So one of the other best decisions I ever made in my life, the first year I arrived in Colorado, I hired Anya Belasova initially as a postdoc. And we knew that if we ever wanted to take this technology to the clinic, we had to find a safer way to reprogram. Yamanaka had used viral vectors to introduce the reprogramming factors into the adult somatic cells. And the FDA would never approve viral vectors because they integrate at random. They could potentially cause an oncogenic event. So Anya convinced me several years later to actually hire her husband, Igor Kogut. And together, and this was really, we heard perseverance in one of the videos of one of the summer interns. This was really a story of perseverance. It took about 6 years to figure out how to do this. But they've developed an amazing, very efficient technology that doesn't use viral vectors. It doesn't even use DNA. It uses messenger RNA or microRNAs. And if you remember, these are labile. They don't persist. They have a very short half-life. So it's a transient exposure to the RNA that actually encodes the reprogramming factor. So they discovered a combination of 6 different factors that they could introduce. And when they introduced them in combination with mature microRNAs, it increased the reprogramming efficiency tremendously. Anya could actually even reprogram single cells, which was unheard of. So using that technology, then they were actually able to. So back up 1 second. So one of the diseases that one of these blistering diseases that we focus on now is one of the recessive disorders, recessive dystrophic EB, which again is the loss of the structural protein collagen 7. Dystrophic EB is probably the cruelest form of EB because the patients can actually live into adulthood. And if they do live into adulthood, most of them will actually succumb to very aggressive skin cancers, squamous cell carcinomas. And you can see that there is no cure. They blister, they scar, they're in essence constant pain. And so this is the disease that we have decided to work on. And using Anya and Igor's technology, what they've been able to do is actually dramatically reduce the time it takes to make gene-edited, reprogrammed, induced pluripotent stem cells, or iPS cells. So initially, if you look at the schematic on the left, the plan was to take a biopsy, put the fibroblast in culture, initiate reprogramming, isolate iPS cells, then go through genetic correction using gene editing technology like Cas9, select out correctly edited clones, expand those, and then differentiate them into adult keratinocytes or epidermal cells and transfer them back to the same patient. Well, that's a very long process. And when we first initiated discussions with the FDA said it's too complicated, it's too many steps, too many weeks in culture. We're afraid you're going to introduce spontaneous mutations. So because of the high efficiency of Anya and Igor's technology, they can now actually combine gene editing and reprogramming in a single step. So they initiate gene editing as the first step, and then 2 days later, they transfect in the RNAs encoding the reprogramming factors. So now what used to take months, we can actually get cloned, gene-edited iPS cells in 2 weeks. And so this is the technology that we're using to take forward to get hopefully into the clinic. So on the basis of that technology, we were actually fortunate to form a consortium with teams at Stanford, Tony Oral's team, and Columbia, Angelo Cristiano's team. We were able to get funding from 3 different foundations, the EB Research Partnership, the Sohana Research Fund, and the EB Medical Research Foundation on the West Coast. And we really took advantage of initially starting the consortium in 2016 to then get funding from the NIH. Tony got funding from CERN, the California Institute of Regenerative Medicine. We also got funding from the Department of Defense and several other foundations. So we had initially planned to treat patients with what we call epithelial sheets, where we grow the keratinocytes on a matrix that will support the growth of keratinocytes, and then transfer that sheet onto areas that are prone to blistering. That is actually a very time-consuming, expensive process. And recently, within the last year, we've decided that there's a better way to deliver the cells to patients. And that way is to use a device developed by Evita Medical. It's called a Sprayon Skin device. Basically what this is, it's actually approved by the FDA as a device. If you look on the left, that's a little sterile kit that's battery operated. With burn patients is what it's approved for now. You take a small biopsy about the size of a pea from an unburned area. Put the biopsy in the first well that's heated. There's an enzymatic mix in there that will dissociate the skin cells into individual cells. In about 20 minutes, you transfer it into the second well where there is a proprietary buffer. Then the third step, there's a strainer that will strain out debris, let the cells pass through, and then you take a 10 ml syringe and you pull that cell suspension up into the syringe. They developed a proprietary nozzle that then you can just spray the cells onto the burned area. So in the image below, you see a young girl that has a scald burn. And then the image on the right there is 10 months after being treated with Sprayon Skin. And you just see just amazing reconstitution of a normal skin, no scarring. And there's another image shown on the next slide, again, of a young woman who had burns on her face. They got an exception to use on a compassionate use basis, to use the Sprayon Skin device. And you can see there a year later, not only is the skin reconstituted, but also her pigment producing cells, the melanocytes, are reconstituted as well. So she regains her normal pigment skin color. So we've entered into a sponsored research agreement with Evita Medical, and this is the new approach that we're taking to get these cells onto patients. We think it'll be much faster. Will be much less expensive. And when you do this, you take a small biopsy, as I said, about the size of a pea, 1 centimeter squared. When you dissociate it and spray it, you go from 1 centimeter, you can cover 80 centimeters. So that the area covered is much larger than the way we traditionally do with split thickness grafts for burn patients. So finally, coming down to the last few slides, we think we have an approach now to deal with the skin problems, but we know that these dystrophic EB patients also have fragile internal epithelium, like the esophagus is also fragile. So they develop strictures of the esophagus, and these have to be surgically removed to allow the children to be able to swallow and eat normally. Many of them have to be on feeding tubes, especially when they're young. So how do we deal with the internal epithelium? So I was giving this talk at Harvard a couple of years ago, and at the end, I ended essentially there saying, we think we've solved the problem with the skin. What about the internal epithelium? Robert Sackstein, who was in the audience, came up to me and he said, I think I can solve your problem. So basically, he had been studying how, when we do a bone marrow transplant, how do blood stem cells, hematopoietic stem cells, know when you give them IV and a infusion, how do they know to home into the bone marrow? So it took him about 20 years to work this out, but basically he discovered an enzyme that decorates a protein on the surface, and that becomes a ligand that then binds to a receptor that's expressed in the bone marrow. So we initiated collaboration with him. What we basically did is we found a different way to introduce the modification. So to test that, we have a mouse that expresses RFP, or red fluorescent protein, in all tissues, and that's the mouse that we're going to inject into. And we take mesenchymal stem cells that have been isolated from a mouse that expresses green fluorescent protein. We modify the cell surface of the MSCs, and then we inject them into the tail vein of the mouse, and then we induce inflammation in the ear with a chemical. And so the next slide, hopefully if it works, and there you can see it. So this is a blow up on the far left. You can see little windows of the mouse's ear where we've induced inflammation. And the little boxes, there's the three different boxes. And box one, if you look, is where we can actually image, because the ear is thin, we can image in real time. And we can actually see the green cells go to the site of injury. They stop, and then they extravasate out of the blood vessel into the injured tissue. So I'll play the movie again so you can see it here. Well, let's just go back. There you go. You see the cells stop, and then they migrate out of the blood vessel into the tissue. Amazing. The first time I saw this, I was running up and down the hall, and I saw Jill and Carmen, anyone I could find, and said, "You've got to come and see this. It's unbelievable." So the interesting thing was, that mouse was still alive. We took it back down to the mouse house, and we decided to bring it up 5 weeks later and see, are the cells still there? And in fact, the cells were still there, and they had actually multiplied. And you can see this is 8 weeks post infusion. So to mimic how we might use this in the clinic, we decided to do multiple dosing, because I think we'll have to do multiple dosing with patients. So we gave 4 doses once per week, and then we imaged. And what you see is that the cells actually accumulate over time. So we're really optimistic that this may be a way. We know that mesenchymal stem cells actually synthesize type 7 collagen. It's secreted. So we think these modified cells will go to sites of injury, of skin fertility or epithelial fertility, and they actually become a little factory to make the missing protein, because type 7 collagen is secreted. So that's the approach that we're working on to see if we can improve even further as a way to treat the internal epithelia. So I'll just end by saying that unfortunately, since you weren't on campus, the interns usually get to tour the GMP facility, our Gates biomanufacturing facility. In order to move any of these therapies into the clinic, we have to have what the FDA calls a good manufacturing practice facility. So we were very fortunate. It was a real community effort. We raised money from the private sector. We partnered with the School of Medicine, University Hospital, Children's Hospital, to build a facility that was opened in 2015. It's just across the street from our research labs. We now have a staff of 43 people over there. We can not only manufacture cell-based therapies, but we also have a protein manufacturing suite with a large bioreactor that you see there, where we can actually make biologics or proteins for under GMP conditions as well. So I mentioned last night when we had the the celebration of what normally would be Charlie's Picnic, that we call it our biomanufacturing facility, the GBF for short. Just 2 weeks ago, literally, were able to complete a major milestone where the first cell-based therapy, this is a CAR T-cell based immune therapy for cancer, where we take patients' T cells out, we genetically modify them in the manufacturing facility so that they express now a chimeric receptor which will bind to the surface of leukemic cells and kill those cells. So we genetically modify them, expand them, and then infuse them back into the patient. So for the first time, we did all of that on campus. So the protein, the cells were engineered in the GBF. This was a clinical trial that was led by the CU Anschutz campus. And then there was a patient at the University Hospital that was treated with this cell-based therapy right on our own campus. So we're really pleased to see this now go for a full circle. So I hope I haven't gone too long. Jill, I didn't see her screaming at me, so maybe I'm okay. Sorry for the delay in getting things going at the beginning. If there is time for a question, I'll be glad to try to address it.
Thank you, Dennis.
Thank you very much. We have time for some questions. I encourage the interns to ask some questions.
I have a question about the Evita device that you were talking about. So I noticed that in the diagram it said that the surgeon does the biopsy. So is this like an inpatient treatment or like an outpatient treatment, or do they do it at home? Like, how does that work?
Yeah, I mean, initially it will be inpatient, but clearly it's such a simple way of taking the biopsy and then delivering with the Sprayon Skin device. Almost anyone could be trained in how to do that. So what we actually envision for treating these EB patients, Anya and her team are working to see, can we freeze the keratinocytes after we differentiate them from gene-edited iPS cells? If we could freeze the keratinocytes, then we could actually ship these. So anybody, even in a remote location, could actually take a biopsy, fly it to us overnight. We would put the cells in culture, do the gene editing, the reprogramming, differentiate them into keratinocytes, freeze them, and then we could just ship the frozen cells back, and they could use the Sprayon Skin device to deliver them and it wouldn't have to be at a major medical center. It could be even in an outpatient setting.
That's awesome. Is there any potential for like rejection of the cells?
No, not these cells, because remember, they come from the same patient that we're treating. I mean, the one thing, it's a good question, because the real problem with the skin is, it is the largest immune organ. So the skin is programmed to fight off infections. So the immune system is very active in the skin. And so you can't transplant skin cells, keratinocytes, from one patient onto another patient that's unrelated. They'll be rejected. So this is the beauty of using iPS cells, where you take the biopsy from the same patient you want to treat.
Cool, thank you.
You're welcome.
Other questions? So I have a question about the dosage you'd have to supply. Does spraying on more cells equal better treatment, or perhaps not?
I mean, we don't obviously know yet, Joe, for EB patients. I mean, with burn patients, clearly they go from a 1 centimeter biopsy, they cover 80 centimeters. And so clearly the stem cells that are in that small biopsy are dispersed, sparsely, but they do have other support cells around them. So there are fibroblasts there. There are other keratinocytes that aren't stem cells. And in that milieu that you create when you do the digest, I'm sure there's something magical about that. And we're trying to figure out exactly the right combination of factors that we need just to add to the keratinocytes themselves for EB patients. But it's really amazing. It's very, it looks too good to be true, but it was actually discovered by a Australian clinician in Perth, Australia, way back in the early 2000s. She, after one of the explosions in Bali, the burn victims were flown to Perth, and she was just overwhelmed. And the patients, they were, the traditional way is you can grow the keratinocytes over 3 weeks, and then you can make enough to graft the entire surface, but the patients weren't surviving. So just out of desperation, one day she decided, I'm not going to expand them. I'm just going to, I think she mixed them with fibrin and just sprayed them on. And that was the genesis of it. And, now that you actually have, especially with darkly pigmented individuals, you have melanocytes in there as well, and they modified the media to preserve the melanocytes. So you get beautiful reconstitution of pigmented skin as well. So there's lots of applications. The company is initiating a trial for vitiligo patients, which is an autoimmune disease where you lose your pigmented cells. And hopefully Colorado will be one of the sites for those patients, because there's currently really no effective therapy. You can do UVB exposure, but sometimes that works, sometimes it doesn't. So I think there are going to be many, many applications of this technology.
I have another question. So you mentioned autoimmune diseases, and I first of all, this was really fascinating. Thank you very much. I wanted to know if you see any potential for this to be used on internal organs as well, the linings of internal organs. There's research that I'm interested in conducting, specifically towards people with ulcers developing on internal linings of organs. So I wanted to see if you see any parallel that could be translated over to that, or if this is really different.
Yeah, absolutely. There are, obviously, we have ways of getting small micro devices into internal epithelia. So certainly, the EB kids have problems with the oral cavity. So I think easily you could spray into the oral cavity. But I think the same goes for, airway epithelium, intestinal epithelium as well.
Thank you.
All right, any other questions? All right, well, let's thank Dr. Roop.
You're welcome.
Thank you, Dennis. It was wonderful, as always. Even if you've heard it about 80 million times before, you always learn something else.
Thanks, Jill.
Excellent. So to wrap up here, we can't do this without the support of everyone involved, including our wonderful donors, our mentors, the leadership of the program, the staff that runs things, and of course the interns. So thank you all very much. For those, the interns and the program staff, I'd ask you to stay on the call, and everyone else, again, thanks for participating today.
We loved having you.